Today's topic is CRISPR for genome engineering implications and challenges. And we have an eminent speaker for the same. Before we start our webinar, I would like to introduce our moderator, Dr. Cletus, Assistant Professor, Department of Biochemistry, who has completed his PhD from prestigious JIPMER and has been instrumental in shaping the central research lab at our institution. And we have also Dr. Riju Matthew, Assistant Professor, Department of Biochemistry, Believers Church Medical College. He is a terrific professional when it comes to lab activities and shows exemplary leadership in running and improving our central lab service. Without much ado, may I request Dr. Cletus to introduce um, our international guest speaker for the day and go ahead with the proceeding. Dr. Cletus, over to you.
Yeah.
Thank you.
Yes. Well, thank you very much, Lynn. It is indeed my proud privilege to welcome Dr. Sijo Chemanur to our 58th international webinar organized by Believers Church Medical College Hospital, Tiruvalla. Dr. Sijo completed his post-graduation in biochemistry and molecular biology, and he was the university rank holder and gold medalist from Central University, Pondicherry. Subsequently, he was awarded Junior and Senior Research Fellowship from Council for Scientific and Industrial Research, and also a Junior Research Fellowship from Indian Council of Medical Research. He pursued his PhD from National Center for Cell Science, NCCS Pune, a premier institute of Department of Biotechnology, Government of India. He carried out postdoctoral research studies at UPMC Hospital, Pittsburgh, Pennsylvania, United States. Later, he joined the Fox Chase Cancer Center, Philadelphia, as postdoctoral research associate. In October 2020, he joined as formulation scientist 2 at Precision Nanosystem, Vancouver, Canada. He has extensive experience in genetic engineering, and currently he is into indigenous vaccine development program against SARS-CoV-2. Today he will be talking about CRISPR-Cas9 technique, which earned Emmanuelle Charpentier and Jennifer A. the 2020 Nobel Prize in the field of chemistry. We wholeheartedly welcome Dr. Sijo for sharing your valuable insights about this topic. Over to you, Dr. Sijo.
Okay, thank you. Thank you for your kind introduction. I will share my slide. 1 minute. Okay, can everybody see the slides?
Yes, sir.
Yes.
Yeah, so I would love to start. I'd like to start the presentation with a change of my title, because I know that most of people are in the medical field. So the title, the modified title will be CRISPR-Cas9 and its implications in medicine, because it's a very broad topic. I cannot give justice if I cover all the topic and limitations. So the title of my topic will be CRISPR-Cas9 and its implications in medicine 102. There is a reason for 102 because it's not 101. I assume that most of the people know the basics of molecular biology. That is, what is a DNA, what is a RNA, what is a protein, what is a gene. Without that, it might not be good to move forward, because I hope everybody knows this basic molecular biology facts to move forward. So that's why it's named as 102. So the outline of my talk will be introduction about CRISPR-Cas9, history of CRISPR-Cas9 gene editing technology, basic science to bedside treatment, applications in modern medicine. So what are the different therapeutic strategies people devised for on basis of CRISPR-Cas9, and the conclusions. So before going forward, if anybody have any doubt, I don't, you can directly ask me, unmute and ask me and stop me at any point of time. So when you first think about CRISPR-Cas9 or genetic engineering, this things comes to my mind. It's about movies, X-Men or Spider-Man. There are genetically engineered mutant humans or different animals, which comes to our mind with different fantastic movies. And especially when you think about Spider-Man, you can see that a spider will come and bite a person, the hero, and then you can see big animations that it goes inside the gene, there's a change of the DNA, some stretch of DNA is removed, new stretch of DNA is inserted. This is what exactly even the CRISPR-Cas9 is doing. So this is how the CRISPR-Cas9 works. So a cell is transfected with an enzyme complex containing the guide molecule DNA cutting enzyme. So you have a guide molecule which guides the DNA cutter to a specific target. So on the step 2, this guide molecule guides the DNA cutting enzyme to one particular target. And in the third step, the enzyme cut the DNA target. And in the fourth step, this target DNA strand is removed and the new changed DNA strand is attached. This is the fundamental basis of gene editing. So most importantly, it necessitates the role of a guide molecule, which guides the target of editing. And the second is the DNA cutting enzyme. In this case, it will be Cas9. I will explain that in detail, but you can think of an analogy as a rocket or a missile. So the rocket in a missile is the target molecule or the guide. The guide targets it to one particular location. And when it reaches the location, it explodes or the bomb explodes. Or in this case, the Cas9 genes activate and is cut the DNA. That's how the analogy works. Think of a missile and that's how basically it works. So again, I'm telling it need two molecules. Most importantly, it need guide molecule, which guides it to a particular location. And secondly, there is an enzyme, which is the DNA cutting enzyme. In this case, it's Cas9, which cuts the DNA. So keep that in mind. It's very important moving forward. So what is CRISPR? CRISPR is clustered regulatory interspaced short palindromic repeats. It's a big name. It's a complicated name. You don't have to remember it, but it's a short repeats of sequences interspaced by some spaces. And what is Cas9? Cas9 is CRISPR associated protein 9. So I'll explain that in detail. You don't have to remember this. This is just to give you an understanding about what CRISPR means. So what exactly is this CRISPR sequences? So CRISPR is a family of DNA sequences. The first thing we should understand is CRISPR is just some DNA sequences. It's just some DNA sequences found in the genome of bacteria or prokaryotes. They're derived from the DNA fragments from bacteriophages, which infects prokaryotes. It's just like, it's viral DNA sequences inserted into the prokaryotes. It looks complicated, but we will look into it together in future slides. So this is how the entire story of CRISPR story came about. It's a serendipity. That means people were looking forward to some other genes, but suddenly they found out that there is something else in the gene. So they were looking for this particular gene. In this case, it was alkaline phosphatase isozyme, a conversion enzyme in Escherichia coli, E. coli from Japan. This group is from Japan. Atsuo Nakata group from Japan. And when they looked into the sequence, especially at the C terminal, they found an unusual structure was found in the 3' end, that is after the gene of interest, 3' end of the gene, that is end of the gene. They found 5 highly homologous sequences of 29 nucleotides were arranged as direct repeats with 32 nucleotides as spacing. So this is the first report of any CRISPR sequences. It reported in 1987 by a group in Japan. So after that, there were a lot of research going on in this field. In 1993, a group from Netherlands found out that this kind of repeats are also present in Mycobacterium tuberculosis, which causes the tuberculosis. So they repeatedly found the sequences in Mycobacterium. Again, into the 1999, this group from Spain, Mojica group from Spain, found out the similar repeats in various strains of bacteria, from RK bacteria, from E. coli, from Streptococcus typhi. A set of bacteria, they found this similar sequences. So that was interesting. And so he called it as short regularly spaced repeats, or SRSRS, short regulatory repeats. And in 2002, Jansen's group, Jansen and Scholl's group from the Netherlands, they found out that these sequences are not only just some sequences, but it is coincided or associated with some specific genes. So he observed a homologous genes. He called it as CRISPR associated systems, or Cas, accompanied by these repeated sequences. So Cas protein showed to have helicase and nuclear nuclease motifs. And this is the first time he used the acronym as CRISPR in this report. So CRISPR word is first used in this report in 2002. Moving forward, you can hear me? Can you hear me?
Yes, sir.
Unmuted. Kindly please mute.
Okay, good. Everything good? Okay. So moving forward, so till now we saw that there are some sequences in bacteria. Some people reported these are clustered in a group of bacteria. Other person like Jansen reported that it is associated with some genes, and he called it as CRISPR and CRISPR associated genes, or Cas proteins. And this group, they published in 2007 in Science, the Rudolph Barango group, they published that it is not only just some sequences, or it's not only just some genes or proteins. These are very specific proteins important for viral resistance in bacteria. So that means if there is more similar sequences, that is causing some viral resistance for the bacteria. So it's causing some immune resistance against bacteria. It's not just sequences, it is very crucial for resistance against viral infections in bacteria. So there are some reports for, in this case, there are two sequences, S13 and S14. If you have the sequences in a bacteria, it can stop the infection of 858 phage, bacteriophage, and 2972 bacteriophage. So the infection rate sensitivity is very low. So this means that whenever you have the sequences, the bacteria have resistance against the virus. That is this paper. So it's good. So we reached a point where people have identified the short sequences, people have identified the genes associated with the sequences, and now we know that these sequences have some specific functions. So till now, everybody clear? Any doubt? We have to understand at least this side, because otherwise it will be very difficult to move forward. It's getting more and more complicated, and it will be more tougher and more complicated later. So at least this part, all people should be very clear that there is some sequences in bacteria, there are some proteins associated with it, and these proteins and DNA are important for viral infection and resistance towards viral infection in bacteria. So what is the need of CRISPR sequences? It's just like a bacteria is telling a virus, we have our defense. Don't mess up with us. It's as simple as that. So it has the bacterial defense system recognizes these foreign sequences and acquire resistance to chew up these sequences. used to detect and destroy DNA from similar bacteriophages to prevent future infections. It's same like how a vaccine works. So if you have a memory of a DNA, it can store the DNA, it can produce proteins against that DNA and chew it up. That is what the CRISPR and Cas9 protein in bacteria means. Okay, so CRISPR associated protein 9. Why so much fuss about CRISPR9 or Cas9 protein? So this Cas9 is one of those protein which is associated with CRISPR. There are Cas1, Cas2, Cas3, similar proteins till Cas13, Cas14, all those kind of different proteins. But Cas9 protein belongs to one particular subtype of group which functions very differently from other proteins. So I will explain that in the future slides. Just think that Cas9 is one of the important protein within that clustered sequences. It's a very important gene which transcribed into this protein. So this is an enzyme used for CRISPR sequences recognition and cleave DNA. So if you look at the right side of this slide, you have a big structure of this protein. It has something called guide RNA. It's written as guide RNA. So guide RNA is something I talked about before. It is important for target acquisition. You put a missile from Vancouver to Kottayam or Tiruvalla, it should reach there. So the guide RNA is something which take the Cas9 to reach to Tiruvalla. So it's the guide which is guiding the protein. And the target DNA is the place where it should go. So it is going to Tiruvalla and attacking and cleaving the DNA. So that's where it functions. It has different domains for guide RNA, target DNA, and all these different domains, helicases and nicases domain, or which are domains needed to cut the DNA. So basically you fire the rocket missile by guide RNA take the Cas9, it goes to the target and cut the DNA. Clear, everybody? It's not
Yes.
complicated. So moving forward, so this is the main mechanism of CRISPR-Cas9. So in bacteria, what happens is when a bacteriophage or virus infect the bacteria, it leaves this DNA inside the bacteria. So the blue one is the phage DNA or viral DNA. So what this bacteria do is something called spacer acquisition. That means it takes the small part of the viral DNA and incorporate into the genome of the prokaryote or the bacteria. So what bacteria does is it takes a small part of this virus and incorporate into the DNA, into the genome. So the next phase is called CRISPR biogenesis and processing. That means this region is transcribed. It is converted from DNA to RNA and it is processed. So this is the premature RNA, this type. It's processed into small RNA. It's also known as the CRISPR RNA or CRRNA. It's written here, CRRNA. And this CRRNA is identified by different subgroups of proteins. You can see here there is a Cas3 protein and other proteins on the type 1 group, Cas9 protein in the type 2 group, and there are different proteins including Cas13 and 14 and other groups in the type 3 subgroups. So in this case of Cas9, it necessitates two RNA. One is the CRISPR RNA, another is trans-activating RNA or tracer RNA. I will explain about that a little later. Just imagine that for activating the Cas9 protein, you need two RNAs. One is the CRISPR RNA and other is the trans-activating RNA or tracker RNA. Okay, all good? So again, the mechanism is virus infecting bacteria, bacteria taking up some portion of the virus, incorporating into its genome. It's then transcribing into small RNA, identified by the group of proteins called Cas9 protein or Cas proteins. Cas3, Cas9, and Cas13, 14 protein. And what happens after that? So these complexes will redirect into this genome and cut it, chew it up. So these complexes will go back to the viral DNA and chew it up. So basic function is to chew up the viral protein after its detection. Make sense?
Yep.
Make sense?
Yes.
Okay. So that is the main function of the CRISPR and Cas9 protein. But the groundbreaking finding is done by Emmanuel Charpentier and Jennifer Doudna. They showed that this Cas9 protein is really programmable to specific DNA sequences. That means if you take the CRISPR RNA inside a human body, it can direct into its target and cut. So they showed that in the science paper published in August 17, 2012, they showed that it's not only functional in prokaryotes, you can take that protein, put into your body, and tell them to cut anywhere you want, and it will go and cut. So that in the right side of this slide, you can see in this, if you look at this panel, this lane, it has a linear plasmid, it has the Cas9 protein, it has the CRISPR spacer 2, that is CRISPR RNA 2, it has the tracker RNA, that is transactivating CRISPR RNA, it has the magnesium, and it cuts the DNA. So this is the band around 6 kb, and it is cut into maybe around 4 kb and then 1.5 kb. So it can specifically cut a particular target if you add a Cas9 protein and a CRISPR RNA along with the tracer RNA. So it is a programmable protein, similar like a restriction enzyme, but it is more like more than a restriction enzyme, because we can directly tell them go and cut this place, and it will cut it for you. So this is the very seminal paper in the field of CRISPR Cas9, and for this they got this Nobel Prize. For this finding, the Emmanuel Carpentier and Jennifer Doudna got Nobel Prize last year for that paper. So it's one of the most seminal paper in that particular field. Moving forward, there are a lot of applications of genetic engineering using CRISPR-Cas9. I give the list of references if anybody want to go into the details. It includes gene surgery, drug development, animal models, genetic variation, materials, food, fuel, all the different kinds of industry you can exploit using biotechnology. But today for our discussion, I will concentrate only towards gene surgery and drug development, and most of its application in medicine. So I hope you're all medical students. That's what Cletus told me. So I will concentrate my presentation mostly onto this area, how therapeutically these are important. Okay, so there are some requirements for using CRISPR-Cas9. What are the requirements? It needs Cas9 protein, or it's a vector containing the Cas9 gene sequences under various promoters. So basically it needs Cas9 protein. And it's also need the guide RNA. It need a rocket. A missile need a rocket to reach the target. So it need a guide RNA or CRISPR RNA, or it can be expressed by a vector. You put a plasmid containing the guide RNA, it will express the guide RNA in the cells. Okay, all good. So it need two things. One is Cas9 protein, other is the guide RNA. And there next is different delivery mechanisms. We have to deliver that into a cell. You have lentiviral method to deliver all these constructs inside the cell. You can use DNA plasmid and put it inside the cell using electroporation. You can use mRNA, Cas9 mRNA or guide RNA, mix together as an RNA complexes and put it inside the cell, or we can use ribonucleoprotein. That means you can commercially buy the Cas9, mix it with the guide RNA, and put it inside the cell. If it is getting too complicated, stop me, I will explain it. So this is not very, it's not very rocket science, but it's very simple. You need two things. One is the Cas9 protein and second is the guide RNA. You mix it, put it inside the cell in one way or the other. What it does is it breaks the DNA. It's called double-stranded break, DSP. It breaks the DNA at one particular target. So when it is broken, there will be DNA repair. So how the DNA repair itself? It can be either non-homologous enjoining, that means it start inserting some sequences in between, or if there is some homologous DNA present near the site, it can recombine. And this method is called homology-directed repair. So this is very basic recombination method happening in the cells all the time. And these are different ways it can recombine. When there is a cut, and if there is any homology sequences, it directly go and insert. And the HDR, or homology directed repair, is the fundamental basis of knock-in strategies. There is something called knock-in, especially when you want to repair a particular gene of interest. There is some bad sequences and you want to repair it with the good sequences. You need to use the HDR method, that is homology directed repair, HDR. So all good. So there are some scientific procedures needed. The first and foremost and the most important is identification of a target gene sequences. We have to identify if you have to change a particular sequences. So there are a lot of diseases. I'll go into that in detail a little while. But there are a lot of diseases caused by some bad DNA or bad genes. So you have to identify that bad genes to change into a good DNA, good gene. So identification of target sequence is the most important. Second is design of the guide to reach to the target. Third is sketches treatment strategy, especially when you want to treat a particular disease. Fourth is delivery strategy to express Cas9 or guide RNA. There are different delivery methods you want to incorporate. You can electroporate, give electric shock and put the DNA sequences inside the cells, or you can use viral method of delivery inside the cell, or non-viral delivery, which our company works on right now, is to use liposomes to deliver the CRISPR inside the cells. And the fifth is analyze the readout of gene editing. We have to some way identify whether there is any gene editing happened or not. That is one thing. That is the fifth thing. And the last and the final is the functional validation. So if there is any change in the DNA, it should be identified by functional readouts. That means if you have a non-functional gene or bad functional gene, and it is repaired, now it is a good gene. So if it is a good gene, a good protein should express. So that kind of functional readout we should have. So these are the scientific procedures behind any gene therapy. So this is some of the work which I have done in Fox's Cancer Center. So I used CRISPR-Cas9 based deletion of a particular gene called THPOK. The gene name is ZBTB7B and the protein name is THPOK, which is important for T cell differentiation. So I used ribonucleoprotein. So I used Cas9 with the guide RNA, mixed in vitro, mixed outside, 30 minutes incubation at 37 degrees, electroporate into the cell. Electroporation people use all the time. So electroporate into the cell, and this dark band represents the expression of the protein of my interest. And I have done 12 different clones of cells. So I electroporated cultured the cells in single cells in one well, and I collected 12 different clones, and I looked at the expression of the protein by Western blot analysis. So as you can see here, the fourth clone has almost none of this protein. So most of the gene is deleted out in this cell. So that means my CRISPR-Cas9 is going to the target, cleaving that protein, and that gene is not functioning now. So therefore, I move forward with using this clone for my future works. And I did the same thing in human CD4 T cells. I used this guide RNA, sgRNA is called guide RNA, and I deleted out the protein in the human CD4 T cells. So this is doable. This is not something astronomical or something. We can do it very efficiently in the lab. So there is ex vivo gene therapy people are doing. Ex vivo means take out the cells from a living body. So it's outside the body. So people use this as a most efficient method now for gene therapy. So you take out the target cells, especially hematopoietic stem cells or the T cells, culture it in vitro, edit the gene, change the gene, retransplant the modified cells into the body. And that causes a lot of disease remedies, including leukemias, including lot of like sickle cell anemias and a lot of other diseases and HIV. I will explain it one by one. So this is first is what is our applications in HIV? So there are 3 papers published in different journals, PNAS and other journals, which targeting CCR5. So as a medical student, you must be knowing about what a CCR5 is. Can anybody explain what is the function of CCR5? Anybody? CCR5? No? Anybody can unmute? Nobody? Okay. I'll count 1, 2, 3. 1, 2, 3. Okay, CCR5 is important for the entry of HIV into the T cells. So CD4 T cells express CCR5, and human immunodeficiency virus HIV enters the T cells using the CCR5 receptor on its surface. So what these people thought is, if I delete the CCR5, HIV cannot enter. So what they did is they targeted CCR5 gene and they deleted the CCR5 gene. So there is no CCR5 gene on the surface of T cells. HIV cannot enter, and that's how the T cells are more resistant towards HIV. Is it clear? Okay, so that is one strategy people are using now. So it's called CCR5 disruption in iPSCs, induced pluripotent stem cells. In this case, it's in primary CD4 T cells. Gene editing of CCR5 using adeno-delivered CRISPR-Cas9. So they design a guide RNA to go and bind to the CCR5 gene and delete it. And delete at the 5' end or before the gene. So the gene cannot be transcribed or gene is deleted out, and the T cells cannot have the CCR5 receptor and HIV cannot infect. That is the idea. Simple. And this group from Philadelphia, very close to where I did my postdoc, what they did is they injected this vector. So this is the vector they injected. This is an AAV vector, that is adeno-associated virus vector, and it has the guide RNA towards many viral genes, including LTR, GAG, POL, EMV. So GAG, POL, EMV are all viral genes. So they designed guide RNA to target those genes. So that means if you have a viral infection, for example, if anybody have a HIV infection, you can get this injection, and this Cas9 and CRISPR and the guide will go and cleave those genes of virus inside our body. It makes sense? So what they did is they injected this virus into the veins of monkeys, and so the Cas9 and the guide RNA went to the target cells and deleted out the viral genes. That means if you have an HIV infection, you get this injection and you can delete all the viral genes from the body. It's fantastic. They got it successfully in mice. It published last year in November 2020 November in Nature Communications. It's a very big achievement. They can make a person HIV negative by this method. Going forward, next is a very other hot topic, which is cancer immunotherapy. I hope many people are interested in this field now. There's a lot of interest going forward to the cancer immunotherapy. It's all centered towards PD1 and PD-L1. So PD1 is a protein is expressed on the T cell surface. And whenever there is a PD1 protein on the surface, it causes the T cell to get tired. Like many students are tired already now. The T cells are already, it makes the T cells tired. So when the T cells are tired, it cannot function, it cannot kill the cancer cells. So what people have done, people have designed something to delete this PD1 gene. So it causes the PD1 gene deletion. So they designed some targets on the PD1 gene. They designed the target and designed an HTR which causes the stop the PD1 gene transcription. So basically it is stopping the PD1 expression on the T cells. So because there is no PD1 on the surface of T cells are more active, it can kill more cancer cells. That is the idea. And anybody heard about CAR T cells, CAR T cell immunotherapy? Chimeric antigen receptor is one of the modern techniques people use to treat the cancer. So what they do is they can make an antibody on the surface of T cells which go and bind to a cancer cell and kill it. Direct targeting of cancer cell is called chimeric antigen receptor T cells or CAR T cells. So this paper they published in Nature paper. What they did is they targeted guide RNA against TCR alpha locus. So, and the basic idea is they deleted the TCR alpha locus and it caused the induced response of CAR T cells. They made a CAR T cell with the deletion of TCR alpha locus. And because of this, as you can see here, the blue graph, the mice with blue graph survived better. That means TCRA is a deletion of the T cell receptor alpha. When you have a deletion of this, the mice survived better. That they published in the Nature. So it's fantastic news. And next, it's in paper, it's in clinic now. People are using this method against PD1, targeting PD1, for example, lung carcinoma. I have one slide for that in later slides. And the other strategy is multiplexing. So they use multiple targeting. For example, TCR alpha, B2 microglobulin, PD1 all together at one time and make the CAR T cells. Those are more functional. That is 10 times more functional. So these are different strategies people use to promote the cancer immunotherapy. So as you can see here, this mice, double knockout CAR T cells, all of the mice survived compared to all these mice. They're all dead. So that's what's shown here. This blue line, that is this one, a double knockout CAR T cell. In this mice, all the mice survived, but in all the other group, they died. So if you translate that into humans, if you have a cancer, you edit the T cells, inject the T cells back, and that human survive. All the other humans die. So it has a direct translational research in translational efficacy in patients. So next is many rare genetic disorders. This is Duchenne's muscular dystrophy. People use this group use fibroblast, and in the cells stems made stem cells out of it. There is a change in the outer frame of this gene, dystrophin gene. They changed with specific guide RNA, and they made it in frame. So this gene was out of frame. They made it proper. That means they deleted a particular portion of the gene and made it proper. So they changed the gene, made the bad gene into a good gene, and now there is no disease. So as you can see here, the blue graph, the second panel, the blue, the green circles here express the functional dystrophin. In the DMD patient, there is no dystrophin, very less dystrophin. And when it is reframed or repaired, it started expressing good dystrophin, more green. That means you have a bad dystrophin in Duchenne muscular dystrophy patients. You do genetic repair, and now you have good dystrophin. That means the people can survive. Next is postnatal. So they have a mouse model of muscular dystrophy. It's called MDX. They have mouse paralysis, and this mouse die naturally. This is a mouse model of dystrophin gene mutation. So they treat this mouse with a particular Cas9 and guide RNA. It go inside, and now this mice survives. So it's direct evidence of a scenario of life and death. So this is a fantastic system to work on this aspect. And in the mouse, it's happening very efficiently. Now you have CGD, that is chronic granulomatous disease, and it is caused by a gene called mutation, the gene called CYBB or GP91. So this gene is targeted by this group of people. They showed that they electroporated and they cultured all these genes, all this editing they have done, and they found that the corrected one, the GCD1 and GCD2 are two different patients. And in this, the good proteins are not expressing, but in the corrected C4 and E4, this protein started expressing, good protein started expressing. So they changed the DNA into a good DNA by inserting the HDR or homology directed repeats. And now you have a functional CYBB protein, and the disease can be cured. And the next methodology, we have a lot of examples. I'll just go through one by one very quickly. So in this is neonatal diabetes mellitus, our diabetes. So there is a one base pair mutation at ATA. ATG is changed to ATA. ATG is important for any start codon. So if there is no start codon, gene cannot transcribe. So ATG is very crucial for the start codon. In this scenario, this G is changed to A. So this can be repaired by targeting this particular guide RNA. So guide RNA is targeted here, and it is single-stranded RNA is replaced. So the ATA is changed to ATG. And as you can see here, in the patient, there is no insulin, no red color here, and corrected, started secreting insulin. So these pancreatic cells, they started secreting insulin very efficiently when it is corrected. This mutation is corrected. This is a very typical example of targeted gene editing. It's similar in the case of sickle cell anemia. This is one example. This is another example at the BCL11A sequences at 58 plus 58 site with GATA1 binding. There's a lot of information, but it's just to have I just put this to have a flavor for you that this kind of research is happening now or happening in these days. And in this scenario, you have a sickle cell anemia, and you can see here the edited one, there is no sickle cell anemia. So you repair the change of the bad beta globulin gene, and now you have a good beta globin gene, and there is no sickle cell anemia. Another example is tyrosinemia. Tyrosinemia. So this is also a genetic disease affecting liver, cirrhosis, and all other bad things. Is caused by this single change G to A, and it can be easily repaired by using the CRISPR-Cas9. And now it causes a exon exclusion, and now this exon is included, and we have a good protein, and that is good FAH protein. It's an enzyme needed for the last step of tyrosine conversion, and we have a good protein, and the gene is good, and it is functional in the liver. And this is in the cystic fibrosis. There is no much medications available in this disease because it's genetic. People die because of asthma and other diseases. So this is the only strategy now. People use the phenylalanine 508 deletions. They target this particular sequences with guide RNA and delete out the sequences. This is another example. It's fantastic example for Huntington knock-in pig model. That means Huntington disease is, there is no treatment for Huntington disease. People die with neurological problems later in life, but they fantastically made this pig without a problem of Huntington. They have this bad DNA, bad gene. They corrected it using CRISPR-Cas9 and guide RNA targeting this particular CAG sequences. They removed 18 CAG sequences with 150 CAG sequences and with a human Huntington protein. And now you have a good Huntington protein in the pigs. And they looked at F0. F0 is the first generation, F1 is the second generation, and F2 is the third generation. So they have till third generation, they have a very good protein and there is no disease. And the pigs didn't die. This is against for Parkinson's, people have reported. These are different therapeutic strategies for CNS, people have reported. And in general, if you conclude what are the different conditions people are trying to target, we have HIV infection, multiple myeloma, melanoma, synovial sarcoma, solid tumors, leukemia, lymphoma, thalassemia, beta cell malignancy, sickle cell anemia, B cell leukemia, all these different diseases, they are all bad diseases. They are all life-threatening diseases. They're all makes people die. And there is not much treatment available for these diseases. And the only way move forward to have any possibility of treatment is to look up for something like CRISPR-Cas9 technology for gene editing. And this is the only way out, and people have to optimize it and use efficiently. That is why this technology is so important, and people are so crazy about this because it affects the life of people, especially when you have somebody who is undergoing this kind of diseases. This is one of the clinical trials people have done for. Can anybody say what is NSCLC is? NSCLC. Nobody?
Non-small cell lung cancer.
What?
Non-small cell lung cancer.
Yeah, it's non-small cell lung cancer. So thank you.
Non-small cell lung carcinoma?
Yes, you're right. So what these people have done is they again targeted for PD-1 gene. They took the normal T cells with PD-1 gene, electroporated with Cas9 and guide RNA, deleted this PD-1 gene. Now the T cells are more functional towards the lung cancer. If anybody is interested in the clinical trials, I hope some of the medical students are interested in the clinical trials. These are the inclusion criteria, these are the exclusion criteria, and all the statistics people are interested in the clinical trials. These are the patients enrolled, number of patients treated, and this is the dosage people have done. and different doses of T cells per kg of human. And this is one of the case study people have reported. This is a 55-year-old female with a lot of PD-1 expression and PD-L1 expression. So this is a baseline tumor. They had a big mass of tumor. And at the 52 weeks, they showed that the cancer is reduced, and with high expression or in the biopsy sample in the high area, there is lot more T cells infiltrated. CD3 expression represents the T cell expression, and CD8 T cells as well as the NK cells. So this shows that after this clinical trials at 52nd week, there is more T cells, lung cancer is reducing, and all because of the change in the CRISPR Cas9 method of the PD-1, targeting PD-1. So this is one interesting fact about CRISPR-Cas9. They say in 2018, one person from China, Jiankui, he made CRISPR babies. He made two babies, twins, two girls with CCR5 mutation embryonically. So that means they don't have CCR5 gene in their genome. So these are called CRISPR babies, and it became a big outcry about ethic reasoning of how to change or how to make a mutated human. It's basically a genetically engineered human. It's basically a small version of X-Men, I would say. So it's a huge outcry, and now this person is in prison in China. So this is something we should really make a balance out of it, because you don't want too much change in your basic genome, because we don't know what are the side effects. There are basically three babies now, two twins, girls, and another baby born in 2019, I think, with CCR5 deletion. We don't know what will be their fate, but we don't know if there is any hidden research going on making new mutant human beings. But this is something which we should all be concerned about and move forward. We should be careful about. So this will be my conclusion. I want to stop boring people. So this is the conclusion of my presentation. CRISPR-Cas9 system is very efficient in precision gene editing. It's a very typical example of basic science to bedside treatment transition. That means some researchers found out some sequences in some bacteria, and they did a lot of research, added on to a lot of research, and now you have something which is really treatable at the bedside. You have a patient who is dying, and now you can treat the patient and make that patient living. And that is mainly because of this basic research which some people have done in some bacteria. That is the beauty of basic science, basic research. So this translation of basic research cannot be ignored or undermined. It's very important to have functional basic research, and this is a very typical example of how a basic science or basic research is translated into a bedside treatment. It has a lot of impact in a lot of phases in current day living, including drug development, basic research. I didn't mention anything about what is its relevance in basic research, because there's tremendous impetus in the basic research. People are making, our lab in Philadelphia, we're making at least 50 or 60 different kinds of knockout mice to study what is the significance of one particular gene in one disease, to delete out the gene and see what is its relevance in one particular disease. Is very important. Development of genetically modified plants and food. I know there are a lot of research going on into artificial meat. So you have meat produced in plants or meat produced in cell culture. No, you don't have to go and kill a beef for eating beef. So those kind of research are going on into the food industry. And there are many areas I didn't touch because I want to focus on into the medical aspect because Cletus told me that more people are in the medical field. So I just want to focus myself into the field of medicine. So it's a very fantastic way to move forward, but only with caution. And if somebody remembers this, with great power comes great responsibility. It's from the same movie, Spider-Man. So thank you for your patience. Thank you for your attention. And I would be really happy if you have any questions.
Hi, everybody. I take this opportunity to thank Dr. Sijo Chemanur for that excellent presentation. Sir, such a vast topic, you have made it very simple for us to understand. We were much excited about to hear from you, and this has been a really wonderful session. I open this forum for questions. If any of you have any questions, please unmute and you can ask questions now.
Riju, can I ask Dr. Siju Chemanur a question? I'm George Chandy. I'm a hepatologist.
Sir,
I was
yes,
interested.
yes, please.
Dr. Sijo, I just wanted to ask you about the progress made in hepatocellular cancer. Initially, there was some work. Do we have anything new that has happened in the area of hepatocellular carcinoma? Do you work in that area?
Unfortunately, I don't work in hepatocellular carcinoma, okay. but in University of Pennsylvania, there is UPenn. There is a group working specifically on HCV and the hepatocellular carcinoma related to that, and they made tremendous progress in mice and in non-human primates. The problem comes when it is translating into human. That's where the major limitation is, because you have to get the ethical clearance, you have to get all the regulatory board clearances. So that is the hindrances now. We have the technology available, but we are skeptical about how to move forward. That is.
Yeah, Dr. Sijo, that was a fantastic lecture. I just enjoyed every bit of it. I was thinking, see, many of our patients with HCC are terminal. Yeah. Yeah. So we have a lot of material, and I'm sure people will be willing to try anything at that. You know, they try a lot of Ayurveda. So why not we try genetic engineering is what I was thinking. And we don't have answers for most of them.
Yeah, yeah. We should think of something like that. But again, it's about how ethically you can do it, because you don't want to repeat some previous episodes of John Hopkins trials, and you don't want that thing happen, you know. That will be terribly bad. So it's a very fine balance of everything. But I really hope that if it can cure at least few people, that's the life-changing thing. It's fantastic. Yeah.
Your work is just amazing. Amazing. I just enjoyed every bit of it. Thank you so much.
Thank you so much, sir.
Sir, I have a question that is posted by one of our audience, Sam Sadar. He's asking, can gene slicing by CRISPR-Cas9 delivery be possible in vivo?
So there are many reports people are doing it in vivo, at least in NHP non-human primates. The study from the Temple University clearly shows that an AAV-mediated gene editing is possible in vivo. So they just injected the AAV virus in the blood, and they changed the gene, removed all the viral genes from the body. So it is possible in vivo, and especially in the case of the CAR T cell, they are injecting many adeno-associated virus into the vein. But the progress is not as expected, but it is on the positive side.
Thank you. I have a question like, this technology seems to be precise. Then if you say precise, the target, the gRNA you use, how precise it can be? That means the genome is so huge that if you want to precisely cut, is that so possible?
It is very much possible. That is the beauty of it. So the thing is, like, you heard about a technique called PCR, right? Polymerase
Yes.
chain reaction.
Yes.
You can specifically amplify a particular gene inside the body with 18 nucleotide bases. The primary use is 18 to 20 nucleotide bases. So if you have an 18 nucleotide or 20 nucleotide stretch in your genome, that is unique. And normally, the CRISPR RNA is around 21 to 22 nucleotides. So you can target precisely at a particular target. It is very much doable. That is a fundamental basis of PCR reaction.
Yes.
Yeah.
And I was just dreaming that in future probably there will be something like a cocktail available with the CRISPR technology that can cure multiple disease with a single.
It is something we should investigate, or maybe people are doing at this moment. We, as a company, we are interested in vaccine technology, and we are interested in multiple vaccine in one shot. Like, you inject one vaccine and you have resistance towards all the coronavirus variants. That is what we are looking into, and I hope we'll be successful in near future.
And you are also talking about approvals. No, how far in Western countries this approval, or is it part of treatment modality in any part of the world?
So that can be depending on the place. In US, you have a different approval authority. In Canada, we have a different approval authority. So, but we have a benchmark. Benchmark is the FDA, right? If you have an FDA approval, most of the other people don't outrightly deny it. So the challenge is to get into an approval of FDA. And once you get that approval, I don't think it will be that much. But having said that, it is not that easy too.
And I have the last question from my side. I heard this CRISPR based on my laboratory Feluda test that was publicly told came in newspapers and all. You didn't mention anything about the possibility of more limited resource testing possibility for genetic using this technology.
Yeah, so before at the starting itself, I was limiting towards only the therapeutic applications of the CRISPR-Cas9, and because there are a lot of field, any area of biotechnology is all the area of any area of biotechnology is influenced by CRISPR-Cas9 because of its easiness. When I was doing CRISPR-Cas9, it was so easy. Cas9 protein is commercially available. You can buy it from any company like IDT or Sigma or any other company. You can order the CRISPR RNA from IDT or any other company. You can mix it, electroporate into cells, and then you just screen, and you can get the knockout deletions. So it is so, because of its easiness, people are so jumping into this. So every field is influenced by this. That's why I wanted to change the topic, because otherwise I won't do justice.
Thank you, Dr. Sijo, for that wonderful presentation. We were really excited about knowing the topic as well as looking at the future prospect. Future way by which we can also explore the things.
I wonder, none of the students asked the questions.
Yeah. Any students? If you have any questions, please raise your questions.
I want the students to ask questions.
Any participants who want to raise questions, please raise. Or you can make the questions posted in the message box also.
Dr. Sijo, to be honest with you, our exposure for our medical students to this topic is very limited. So I was even wondering whether you would do we have something called like the TED Talks, we have something called the Best Talks, whether you would do one for us. You could even record it and send it to us. We will play it at a time where all the students are available, because I think this is a fantastic, what I listened from you was awesome. And I would like our students to get excited about what you are doing. So if you would accept my invitation to give us a best talk on this, the current international level at which gene therapy is, that would be the kind of thought I would have.
Yeah, sure. That will be an honor, sir.
Thank you. Thank you so much. I think it'll really excite our students, because especially when you bring in Batman and Spider-Man and all that, I mean, all those examples really made me understand how you cut and splice and all that. I'm not a biotechnologist. I don't know molecular medicine enough, but I'm sure Cletus should be starting talking about it and teaching us cutting and splicing. Thank you so much, Dr. C.
Thank you. Thank you.
There is a broad question that is raised by one of the participants, Devadaiti Sahu. He is asking that there is any guideline that is set for genetic engineering.
So there are guidelines, very precise guidelines, especially after that CRISPR babies. There was a wide range of panic happened especially in US and other Western countries, that all this like embryonic change of a human or genetically engineered humans cannot be formed, ethically should not be made. So those kinds of guidelines is very much precisely written in Western world now. But basic research, or especially the clinical levels, there is ways we can get approval if your science is good. You should have a good science background. Without good science, the therapy will not work. That is why it's so important to have the basic research to back up your clinical trials. If you have a solid basic background of the science, people will understand and people will give clearance. That's what I think.
We have one more question raised. Is this technology specific for point mutations?
Yes. So many of the examples I showed is for point mutations. Diabetes mellitus. It's a one base mutation that is from G to A. And if you know molecular biology more, you can appreciate that the start codon is methionine. If there is no ATG, no genes can be formed. So in this case, in the neonatal diabetes mellitus, what happens is there is a one single base mutation, and that person's life is in a hell. No insulin, lifelong insulin, no insulin, and because of that, there is lifelong insulin and all those psychological issues, all those mess because of one base mutation. And that means it's the G mutation to A. So if you can correct that point mutation, that's what that paper is all about. Change that point mutation. Now you have a good gene and good insulin.
Hello, sir.
Yes, please.
Hello, sir. My name is Mohammed Paul, and I was actually into machine learning recently. So, is there something being done in this angle, like machine learning and artificial intelligence, how that can contribute to this part? Is something done on related to it?
I couldn't hear you properly.
Because there is an echo. He is asking that ML and IA has been utilized in this field for any improvement or any connected.
Yeah. People have used all those kinds of algorithms to, first of all, target. As I mentioned, the first step is to identify the target bad gene. So there are a lot of machine learning and especially artificial intelligence to use, or bioinformatic tools. In molecular biology, we call everything as bioinformatics, right? So there are a lot of bioinformatic tools used to for the target validation, as well as to design the CRISPR-Cas9. So one of the topic I didn't go through much detail is identification of the PAM sequence, the protospacer sequences, which is important for the targeting. So there are a lot of bioinformatic tools available now, which can be utilized to target this delivery. So all those for designing the guide RNA, you can design the guide RNA according to your target sequences. So there is a lot of bioinformatic tools used in that area. Yes.
So finally, yes, please.
It was clear, sir. It was clear. Thank you.
Finally, can we say that this is a magic technology that has potential for good and bad?
Everything is good and bad, right? You inject a lot of saline in the body, it's bad. So everything is good and bad, but only thing I can say is right now, currently, is one of the better technologies what we have in our hands. And there may be a lot of similar technologies which is hidden in bacteria, maybe, because this came from bacteria.
Yes.
That's why it's a wholesome approach. We should think in a perspective like biodiversity. Like some genes are so unique, which can be extinct in some plants or bacteria, or in some wild animals, or some species of rice, or some species of other wild plants. So this is a very typical example how important our biodiversity is, how important our ecology is in a broader perspective. You cannot, it's a wholesome approach of everything as a whole. So right now, if you ask about CRISPR, this is one of the leading and most efficient technologies currently present.
Thank you. If no participants has any more questions, and we take another opportunity to thank you for your excellent presentation, and we look forward for the more presentations like this. Thank you, Dr. Sijo.
Thank you so much.